Good morning, everyone. My name is Alexis Garbarczyk, and I am very excited to be presenting my summer research on investigating the function of the ACA10 uORF. To begin, I'll be covering the most important concept of my research, the upstream open reading frame, otherwise known as uORF. These are regions that lie before the main coding sequence in the 5' UTR, alternatively known as the 5' leader sequence. They're present in approximately 30 to 50% of eukaryotic mRNAs, which I have on the, I'm trying to figure out, on the left, which I have in the, sorry guys, I have listed in the figure on the left. This lists the occurrence of uORFs in different eukaryotic species, including Arabidopsis, which is what I'm working with. So past research has found these elements to downregulate gene expression, and these elements work by several mechanisms: ribosome stalling, release of the ribosome, mRNA decay, retranslation of the 40S. They're classified into 4 major types depending on the positioning of the stop codon. Type 1, the stop codon of the uORF is independent of the main open reading frame. Stop 2, the stop codon of the uORF overlaps with the main open reading frame. And type 3, the stop codon of the uORF is the same as the stop codon of the main open reading frame. And then we have multi-type. So in plants, uORFs have been found in 24 to 30% of mRNAs. This is specifically 37% for Arabidopsis. Now, while I stated before that past research has demonstrated that these constructs tend to downregulate gene expression, interestingly, it has been found, and I've cited several studies that attest to this, that under certain environmental conditions, including pathogen attack, they can actually increase translation levels. And I'll cite an example from the first paper I've listed. So in this paper, they combined an accelerated death gene, uORF cassette, with the PR1 promoter, and found that after pathogen attack, the plant defense protein, lectin receptor kinase, increased in levels. So a possible explanation for this is that perhaps the stress conditions are changing the translational start site, thereby rendering the uORF inactive. So instead of a typical downregulation of protein, instead it's upregulated because there is no longer the uORF being involved in the transcript. But despite all of this research that has taken place in mostly the past 5 years, these mechanisms are very poorly understood, which is why I seek to figure out how uORFs can regulate transcription and translation, and can this change as a stress response. Introducing the ACA family genes. These are genes found in Arabidopsis thaliana. They are calcium ATPases, which function to pump calcium out of the cytosol. ACA genes are involved in the processes I've listed, but most importantly for my research, stress response. In the diagram on the left, this shows a plant cell with the different ACA genes and their localization circled in red. They're localized in the vacuole, the endoplasmic reticulum, and the plasma membrane. The ACA10 gene of Arabidopsis, which is the gene I'm focused on in my research, is localized in the plasma membrane, and this is a cluster 4 ACA gene. It has been experimentally proven to contain uORFs, which is why it's the focus of my research. Here is the beginning of the ACA10 transcript with several features annotated, including exons, introns, translational start and stop, and the UTR region. I will be focusing on open reading frame 5, which is highlighted in yellow. And interestingly, these uORFs are in the exonic region. So I believe that ACA10 uORF translation may be influenced by temperature. This is due to past studies that I described on the effect of uORFs on pathogen attack. And so I believe that another environmental condition, such as temperature, may also contribute to this mechanism. So as more background research, I wanted to study the general effect of temperature on the ACA10 gene. I have conducted a Western blot and a qPCR to display this. My Western blot used the ACA10 BON1 transgenic line. BON1 is an interacting protein which has been found to upregulate the ACA10 and ACA8 genes. And so the anti-HA antibodies are tagged with green fluorescent protein, and stain to display relative protein expression. Looking specifically at my 3-hour marks, what you can see is that protein staining drastically decreases for the temperature extremes other than at 22 degrees Celsius. So this is indicating that in terms of the ACA10 gene in response to temperature, there's some sort of translational inhibition occurring. And then looking at how I quantified transcript levels, I grew plants at 22 degrees Celsius for 2 weeks, harvested, and then incubated at 22 degrees Celsius, 10 degrees Celsius, and 35 degrees Celsius for 3, 6, and 24 hours. From there, I isolated RNA, and then I produced cDNA using an RT-PCR. And from there, I was able to quantify RNA levels using a qPCR. I've used 3 different primers to target different areas of the transcript. Now, what you can see here is that with increases in temperature, the relative transcript level is actually increasing. This doesn't match what we found for the protein levels, which indicates that there might be some sort of post-transcriptional modification going on, hence why we have higher levels of transcript making lower levels of protein. So this does not relate specifically to the element of the uORF. This was just more general background research, which leads me to my experimental objectives. First, I wanted to discover, do other ACA genes in other species contain these elements, or is this unique to Arabidopsis? Secondly, I wanted to know, does the uORF play a regulatory role in ACA10 translation? I predicted that if I manipulate constructs to remove the uORF, the protein levels will change, and this might have something to do with temperature. To explore my first hypothesis, I completed a bioinformatics analysis. I studied Arabidopsis, maize, and tomato. However, I only chose to include my results from Arabidopsis and maize because maize is studied in our lab, and maize is a monocot and Arabidopsis is a dicot, which makes for good comparison. From phylogenes.org, I obtained a phylogenetic tree of all the Arabidopsis ACA genes and maize-related genes. I've predicted the existence of the uORFs with an online program called uORF Flight, which I will get into more detail on in the next slide. However, I will note right now that I've annotated Arabidopsis predicted uORFs in green and maize with the yellow stars. So I will note that it seems that the predicted uORFs are more clustered together. However, I would need additional research to prove if this actually is a clear pattern or if this is just an observation for this particular tree with these particular species. To continue my bioinformatics analysis, I once again utilized the program uORF Flight to produce 2 tables for Arabidopsis thaliana and corn. And I've listed all of the predicted uORFs for each gene. And I have predicted uORFs for ACA4, ACA11, ACA9, and ACA10. However, this website does have a couple of caveats. For example, the ACA10 gene has 1 listed uORF when there should be multiple. One possible explanation of this is that the uORF Flight program utilizes certain criteria set by the website creators, and they only take into account ATG start codons, although alternative start codons for uORFs have been found to exist. Additionally, there are as many as 83 uORFs predicted for maize, which is highly unlikely. So I can't take this information as 100% accurate, although I do feel it is safe to say that uORFs are likely present in other ACA Arabidopsis members, as well as maize homologs. To address my second hypothesis that removal of the uORF will change protein content and this may be responsive to temperature, I've conducted a dual luciferase assay with Arabidopsis protoplasts. Arabidopsis protoplasts are single cells with the cell wall removed that can test for transient gene expression and promoter activity. And the way the dual luciferase assay works is I am testing for the fluorescence of 2 proteins, luciferase and ranilla. The luciferase gene is under the control of the ACA10 specific promoter, which precedes the uORF or lack of the uORF, the manipulated transcript. The ranilla protein serves as a control, and it is under the direction of the constitutive promoter 35S. So once the fluorescence is observed, a ratio is taken of these 2 proteins to determine relative expression. What I found from my dual luciferase assays is that overnight incubation at 10 degrees, 22 degrees, and 28 degrees Celsius, these were my results. I chose not to include my 10 degrees Celsius results because the low temperatures inhibited promoter activity, so the level of expression was the same as the empty vector background level. However, this is what I found for the plus uORF construct in 22 degrees and the plus uORF construct in the 28 degrees. There is a dramatic increase in relative expression. Similarly, I did a slightly different incubation. I did 22 degrees Celsius for 3 hours, followed by temperature treatments of 10 degrees, 22 degrees, and 35 degrees Celsius for 3 hours. And this time I used a broader region of plus the entire 5' UTR, as well as a minus uORF and a minus 5' UTR. Now, similarly, especially for 22 degrees, there was an increase in relative expression. So I had to ask myself, why is there an increase in expression when previous research has indicated that these are supposed to downregulate gene expression? Well, upon further research, one possible explanation is a phenomenon in yeast known as leaky scanning, where the presence of the uORF can actually upregulate gene activity. And this I will cite from a study conducted by Renz and other authors in 2020. So I can't draw any conclusions based on temperature. The results were a bit difficult to interpret. However, in terms of the presence of the uORF or 5' UTR, it very clearly does lead to a higher level of expression. So to finish my findings, for my first hypothesis, this was confirmed. uORFs are likely present in a wide number of species, including monocots and dicots, and this is specific to ACA genes. For my second hypothesis, I was able to figure out that the ACA10 uORF does increase gene expression compared to non-uORF containing constructs. And I did predict that protein levels would change. I wasn't exactly sure how, but this definitely did not match what I expected, which I find really interesting. I can't expand this necessarily to the temperature applications just yet, but if I were to continue my research, I would look at different incubation times for different temperatures and perhaps even other ACA genes. ACA4 and 11 would be a good example because they're localized in the vacuole rather than the plasma membrane, and they have a very drastic double mutant phenotype. So overall, I would continue trials and expand the applications of my trials to try to further elucidate this function. So now, upon finishing my research, I ask myself, is there something we can do with these? Can we find a greater application where we can manipulate plants to produce more proteins under stress conditions to help them thrive? Well, in short, I would like to think so. Not exactly sure yet. I think this research would be very worth continuing because these uORFs are still enveloped in mystery. And I think this experimentation has raised even more questions for me about how these seemingly magical mechanisms regulate gene expression. And I would like to acknowledge my PI, Dr. Jian Hua, my mentor, Dr. Hyojun Kim, as well as the several organizations who supported me throughout the summer. Thank you.